Hi everyone, it's Dr. Jory. This video is designed for students in general biology who are studying gene expression, the central dogma. I cover the 3 steps of transcription and translation. If you like the video, please smash that like button, subscribe to the channel, and enjoy. What is this going to be? AUG. Okay, so what is sitting in here? That's right, RNA polymerase. How did the RNA polymerase know where to go?
Promoter.
The promoter. So the promoter is like at a nightclub saying, come on into the club. So let's say it's right here. Promoter signals RNA polymerase start here because we have the gene we want right there. And the RNA polymerase does 2 things. What does it do?
It unwinds the strand.
Unwinds the DNA strand by breaking hydrogen bonds between nitrogenous bases. And what else does it do? It's a 2-in-1 enzyme.
It pairs the complementary nitrogenous bases.
It does complementary nitrogenous base pairing. So if we have down here, let's say there is G, C, G, but on the RNA transcript there will be what? C, G, C. What is this part of the DNA called? Template strand. So you're using the template strand to build your RNA transcript. Where is this all taking place?
Inside the nucleus.
Inside the nucleus. So this is your RNA transcript. This part has already been built. So it's going in this direction. What is the part of the DNA called in which you're building your transcript from? The gene. Right? The gene is right after the promoter. So this, let's say from this area, is the gene. And the gene is like the recipe. It's got all the instructions on how to build the protein that you're going to make. Right? So where are we here? We're in the nucleus. If we're in a eukaryotic cell, what if we're in a prokaryotic cell? It would take place in the cytoplasm. So this is called the central dogma. It's to go from DNA to messenger RNA to protein. The central dogma. What is the process called to go from DNA to mRNA? Transcription. And to go from mRNA to protein? Translation in the nation. There's 3 steps of transcription. There's also 3 steps to translation. They're both the same. They're not the same steps, but they're called the same. So what are they? The steps? 1, initiation. 2, elongation. 3, termination. So initiation, we already started over here. That's the promoter. There is something that's right before the promoter. Ta-ta! The ta-ta box. So it would be like right before here. So scientists, when they're sequencing a genome, they can look for this type of coding in the DNA, and that gives them the signal that here comes a gene. Once you see the ta-ta, then you know a promoter is coming up, and then that's going to be the gene right after that. Now, what tells it to stop? The terminator. So the terminator, say it's right here. Terminator says that's it. So initiation, promoter says RNA polymerase start here, start building your mRNA transcript. An mRNA transcript is like a photocopy of the gene. That's why we call it a transcript. The second stage is elongation. RNA polymerase, complementary base pairing, building your mRNA transcript. I should have had an M on there. mRNA transcript. In elongation, you have the mRNA transcript getting longer. It tells you right in the word what it's doing. Make it longer. Once you reach the end of the gene, you hit the terminator. Termination, you're all done. At that point, the DNA double helix will go back to the way it was. The hydrogen bonds between the nitrogenous bases, they will bond again. And then your mRNA transcript, it's known as immature at this point. All right, now that you've finished building your mRNA transcript, you have it here, it's known as immature. There's something that you have to do next. It's called processing. And what do we do for processing? There's about 3 different things we do, right? And you all just said it at the same time. So first thing, well, actually, I don't even know if it's the first thing, but one of the things we do is we put on a cap, a guanine cap, and a poly-A tail. So the poly-A just means it has a bunch of adenine. Poly-A tail. Why do we put that on? Put up your hand.
So is it like by enzymes when it goes in the cytoplasm?
Good. So it protects it from enzymes because it's going to go through the nuclear pore. It's going to come out here into the cytoplasm. And we have RNases there, also known as exonucleases, that could chop that up. So now we've got protection. We have the guanine cap, poly-A tail. We also have regions of the transcript. Some are coding and some are non-coding. So we call the ones that are coding regions, we call those exons. And we call the ones that are non-coding, we call those introns. Which ones do we want to keep before we send it out into the cytoplasm? We want to export the exons. So E equals exons, and I equals introns. Introns are the non-coding ones. So those are the ones we are going to splice. It's called RNA splicing. So we're going to chop those out. We'll keep the exons. We're going to export the exons. And another way to remember that is then we're going to express the exons for gene expression. So then your transcript is now mature. So it can leave. You got rid of all the introns. You only have the exons now. Those are the coding regions. And then, of course, you have your cap and your poly A tail. And now, transcription complete. Then we can get into translation. So now we have finished transcription. That took place in the nucleus. We made our mRNA transcript. We have processed our mRNA transcript. It's now mature. We chopped out the introns, kept the exons, put the poly A tail on, put the cap on, exported it through the nuclear pore. It's now in the cytoplasm. It's likely going to be in what organelle right now? Yes, the rough endoplasmic reticulum, which is a continuous endomembranous system with the nuclear envelope. So we're probably in the rough endoplasmic reticulum now. We could just be in the cytoplasm, though, when we meet up with a ribosome. So the ribosome, it's going to start, it has two subunits. Small and large. Obviously, this isn't to scale. This ribosome is ginormous. It's going to be a lot smaller than that. But for illustration purposes, let's do it like that. So we've got a large subunit here. Now let's go back to translation. Translation, we will just translate the message, the messenger RNA. We want to translate that into what? Into a protein. So we want to decipher the genetic code, and that will allow us to build our polypeptide, which will become a protein. Translation, as we talked about earlier, has 3 steps. Initiation, elongation, the same steps that transcription had. And what's the last one? Termination. So initiation is basically start. So start, we're going to have certain, it's always hard to draw this. I don't really have enough space. But the mRNA codons, these are going to be codons. To get things started, you have 3 nucleotide bases. Those are going to be A, U, G to get things started. So start is A, U, G. And that codes for what amino acid? Put up your hand. Methionine. Your voice is a lot deeper than I remember. Methionine. So methionine is what all polypeptides will start with. And that does require energy to do that. Something to consider is that the large subunit has 3 sites. These sites are going to be where the transfer RNA fits in. The 3 sites are called the exit site, the polypeptide site, and the amino acid site. I like to remember it as Environmental Protection Agency because I care about the environment. You could also spell it APE backwards, The beauty of this is that the names of those sites tell you exactly what they do. In the exit site, that will be where the transfer RNA has already got rid of its amino acid, and it will leave from there. In the polypeptide site, that will be where you see the growing polypeptide chain. So at this point, because you just started, there's only going to be one amino acid on there. And that amino acid is going to be methionine. Now the next amino acid is going to come down into the amino acid site. So let's say the next amino acid, I'm going to throw a codon at you. Tell me what the amino acid is. So let's say the next codon is GAG. So it's going to be a triplet code. And it's going to be GAG. GAG. What is the amino acid for that? Who's got it? Unfortunately, I don't have them all memorized. There's 61. Glutamic acid. So the way that you can figure that out is you just look up, use your mRNA codon chart, and that's going to tell you which amino acid is being pulled down next. Okay, the codons that I've drawn here, it's not exactly accurate because there should be another triplet here. So in total, there would be 9 nucleotides. But anyway, what will then happen is the ribosome, it's like a car driving along the mRNA highway or mRNA road. So it's going to continue driving along, and each time you will have, this is going to shift over to that. So now it's going to have 2 amino acids. Because the polypeptide one transferred, then it's going to go to the exit site, it's going to leave. Everything's going to shift over. This one that has 2, it's going to go over to the polypeptide site. Then a new tRNA that's sitting out in the cytoplasm, holding on to the next amino acid, it's going to get called down and everything shifts over. That keeps happening. It happens super quick, and once the dust settles and you read the entire mRNA transcript, that's the whole elongation phase. Elongation tells you again exactly what it does. You're just making it longer. You're adding all those amino acids to it, and that also requires energy. So make it longer. And that requires energy. So polypeptides can range in size. They could be 30 amino acids long. They could be 3,000 amino acids long. Just depends. All right, moving on. So let's say we finally lined up all the amino acids, and they have to be in the exact order that the recipe calls for. The transcript is like a photocopy of the recipe. Okay, the gene is the recipe. The DNA is like this giant recipe book. So you have your specific recipe, you cook your protein, and then eventually you're going to come up on, let's say it's at the end here, you will hit a codon, and it could be one of 3 different ones. So it could be you are great, you are awesome, or you got it all. Those are all known as, do it in red, stop codons. It tells you right in the word what happens. termination. It stops. Stop, you're done. Now, is there, we didn't talk about this in this video yet, the tRNAs, they have what's called anticodons. So they have 1, 2, 3, 1, 2, 3, 1, 2, 3. Those are complementary to the mRNA codons. Okay? Now, once you hit one of these 3 stop codons, is there an amino acid that goes with that?
No.
No. What does this tell you?
Stop.
Stop, but what is it calling for?
Release factor.
Release factor. Good. Release factor is an enzyme. Whoops, release enzyme. Release factor enzyme. And what that does is hydrolyze. So with the addition of water, it breaks a bond. Hydrolyze the bond between the polypeptide. So you have your big polypeptide here. It's getting a little messy now. Between the polypeptide and the last tRNA. Once that release factor comes in, it's like scissors. So it snips the bond from the tRNA to the polypeptide, breaks it apart, and that signals for everything to disassemble. The large subunit falls away, the small subunit falls away. You're going to recycle your ribosome. Your mRNA is probably going to get just recycled, and you are complete. You translated your message into a polypeptide. At that point, your polypeptide is going to look something like this. And what does that look like?
Necklace.
A necklace. That's right, necklace. Not necklace. If you're a necklace, it means you don't have a neck. What do you call this? Do you call it a shoeless? Or do you call it a shoelace? Shoelace, that's right. So why would you call that a necklace? Or sorry. It's a necklace. It's not necklace. Anyway, here we go. There is your primary polypeptide. So what we're talking now about is protein structure. This is the first level of protein structure. Primary polypeptide. Some polypeptides might be happy at this level. Most will go on to the secondary structure, which is like an alpha helix or a beta pleated sheet. Some of them will go on to the tertiary structure, which is now more three-dimensional. And then some will go on to the quaternary, which is 2 or more tertiaries bonding together, like hemoglobin, for instance. It's messy. There's not enough space. That's the hardest thing about this. There's just not enough space to get it all in there. As the polypeptide is moving outwards in the cytoplasm through the rough endoplasmic reticulum, then it's going to break off from a vesicle attached to the rough endoplasmic reticulum. It's going to travel over to the Golgi apparatus, which is known as the post office. Shipping, receiving, packaging. That is where it will reach its final shape, whether it's secondary, tertiary, quaternary is the fourth one. From there, it's either going to bud off into another vesicle, stay within the cell if it's needed there, or you're going to export it. It's going to leave the cell through the plasma membrane and head to wherever it's supposed to go. That is called gene expression. The whole process called gene expression, we also call it the central dogma, and it includes transcription and translation. Whoa, watch out. Anything else?